undergraduate biology and neuroscience double major here at the University of Massachusetts Amherst. At the university, I conduct research in Dr. Gerald Meyer's lab, specifically studying cortisol, the stress hormone. Hormones are chemicals that are released from cells or glands to send a message to other parts of the body through the blood. Everyone has experienced stress. Your heart rate accelerates, your respiration rate increases, your pupils dilate, and you begin to sweat. This is the physiological mechanism of the stress response, commonly known as the fight or flight response, controlled by the sympathetic nervous system. This is a fast and immediate stress response, where the adrenal medulla, a gland located right above the kidney, releases hormones in response to a stressful stimulus. However, stress is not always short-term in response to a stimulus. Long-term stress is controlled by a different mechanism and different hormones, the hypothalamic-pituitary-adrenal axis, or the HPA axis. This is the stress mechanism that I studied in the Meyer lab. The hypothalamus secretes a hormone which acts on the pituitary gland. This hormone, CRH, then causes the pituitary gland to secrete another hormone, ACTH. ACTH then travels through the blood and stimulates the adrenal cortex to secrete cortisol. Cortisol then acts on various tissues to produce a multitude of physiological responses. Researchers can quantify the activity of the HPA axis through measuring cortisol concentrations in saliva, urine, feces, and even blood. However, these samples are only indicative of the activation of the HPA axis at a discrete time point when the sample was taken. Measuring cortisol concentrations through hair samples gives researchers the ability to measure long-term activation of the HPA axis. The goal of my particular research project is to study the heritability of the human stress hormone cortisol. Specifically, this study aims to explore the relationship between the genetic and environmental differences of cortisol levels in identical twins. In the study I'm working on, in collaboration with Boston University, cortisol levels will be quantified through processing and analyzing hair samples from 5-year-old identical twin participants. The long-term goal of this research project is to determine if cortisol is heritable. This would strengthen the understanding of the relationship between a child's specific personality traits and their mental health by comparing cortisol concentrations and individual temperament of the genetically identical participants. Additionally, knowing whether cortisol concentrations are heritable would enable the prediction of future behavioral or physiological consequences of cortisol concentrations. Determining the heritability of cortisol has profound scientific and social implications. It is important to predict cortisol concentrations because an overactive or even blunted HPA axis due to long-term stress can lead to a multitude of disorders, including cardiovascular disease, fatigue, anxiety, depression, and suppression of the immune system and inflammatory responses. Being able to predict cortisol concentrations will allow clinicians to directly combat the effects of an overactive HPA axis in children through medication, lifestyle change, or behavioral therapy. Thank you.